In our previous segment, we have discussed the first two phases of mitotic cell division, and that was, first of all, prophase, and second one is metaphase. We have seen in metaphase that chromosomes are arranged at the equatorial plane. Now we move to the third phase, which is known as anaphase. What will occur during anaphase stage? Let's see. This is the structure of a cell, something like that. This is the structure of a cell. And what will happen during anaphase? Actually, chromosomal spindle, first of all, chromosomal spindle. We have seen chromosomal spindle during metaphase. Chromosomal spindle starts to pull chromosome towards the pole. Starts pulling chromosome towards pole. Actually, how it occurs, if we draw the chromosome structure like that, here is the centromere. Here is the kinetochore region. The deep portion is the kinetochore region. And this chromosomal spindle starts pulling towards the two end. And what actually happened? This is the region. The chromosomal fibers. We are talking about the chromosome. These are the chromosomal fibers, and chromosomes are arranged something like that. Because sister chromatids have detached from each other and arranged something like that. This is actually the position of the chromosomes. Chromosomes are arranged something like that. So these are the chromosomal fibers. These are the chromosomal spindle. This is the chromosomal spindle. There are also the continuous spindles are also. We found the continuous spindles too. This is the continuous spindles. After the formation of this, or rather the splitting of the chromosome, the centromere also splits, and during the centromere region, another type of fibers arises. This is known as interzonal fiber. This is known as interzonal fiber. Actually, splitting of centromere leads to the formation of interzonal fiber. So, actually the splitting of centromere. The splitting of centromere leads to the formation of interzonal fiber. Next we see the forces required. The force responsible for movement of chromosome from equatorial plate to poles. Force responsible for the movement of chromosome from the equatorial plate towards the pole. There are three types of forces actually. We are talking about, first of all, the contraction of chromosomal spindle. Contraction of chromosomal spindle. This chromosomal spindle contract and the chromosomes move towards the pole. Contraction of chromosomal spindle. Number two, stretching of interzonal fibers. These interzonal fibers, stretching of interzonal fiber. And number C is ATP. ATP is another source of energy which is required for pulling or movement the chromosomes towards the pole. Actually, approximately 30 ATP per movement of per chromosome is required. So ATP is another force responsible for the movement of the chromosome. And what will actually the shape of the chromosome after mitosis? It will be something like that. If the chromosome is metacentric, it appears something like that. In case of metacentric, it appears like V. If it's submetacentric, it will appear something like that. This is for the submetacentric, like L. In case of acrocentric, it will be something like that. It is acrocentric, something like J. And in case of telocentric, it will be like that, I. So that will be the shape of the chromosome after pulling off the chromosome towards the pole during anaphase stage. So centromere splits required to send to poles, resulting interzonal fiber formation. The main features of anaphase are chromosomal spindle starts pulling chromosome towards the pole. Interzonal fiber formation takes place, and this interzonal fiber is required for pulling the chromosome towards the pole. And the forces responsible are the contraction of chromosomal spindle, stretching of the interzonal fiber, and ATP. This is the mitosis, the third phase of the mitosis cell division, or the anaphase. Let's remove to the final phase of karyokinesis, or the telophase stage. This is the final or fourth phase of karyokinesis, which is known as the telophase stage. During the telophase stage, actually the reverse of prophase. Telophase stage, actually the reverse of prophase. If we recall the stage of prophase, during telophase stage, what will actually occur? This is the cell, structure of the cell. This is the cell. And during telophase stage, what will happen? First of all, nuclear membrane going to reassemble or reappear. Nuclear membrane reassembles or reappear. Nuclear membrane reassembles or reappears, something like that. Reappear. Nuclear membrane reappear. This is centrioles. Number 3, chromosomes decondense to form chromatin fibers. That is the reverse of prophase, as I already mentioned. That is the chromosome, that is rehydration occurs. Chromosome decondense and form chromatin fibers. So again the same thing will appear. Decondense to form this type of chromatin fibers. These are the chromatin fibers. Number 3, asterase and spindle fibers, which are formed during prophase stage. Asterase and spindle fibers disappear. Asterase and spindle fiber disappear. Actually, this stage is the termination of karyokinesis. So this stage completes the nuclear division. This stage completes the nuclear division. This is the completion of the nuclear division, which will be followed by cytokinesis. In our next drawing, we can see the cytokinesis process. In animal cell, the cytokinesis occurs by the farrowing process, something like that. This is the animal cell. This is the during telophase stage, what has happened during something like that. And during cytokinesis process, what is actually happening? This furrow or the invagination gets deeper. This furrow or invagination gets deeper. And this nucleus will appear more prominent. And finally, two cells of similar shape, and the most important thing is equal number of chromosomes will occur. This is again the same thing. This is the nucleus. This is the chromatin fibers, centrioles rather. These are the daughter cells, which are 2n. That is equal number of chromosomes than that of mother cell. So mother cell or the parent cell 2n. From that, two daughter cells are produced having 2n, similar number of chromosomes. That is why it is known as equational division. This is also, mitosis is also known as astral division or amphiastral mitosis. Amphiastral cell division is also known as astral division because the formation of astral rays. So this is the karyokinesis. After karyokinesis, cytokinesis occurs, and these daughter cells are produced after cytokinesis. This is the total division of cell during mitosis process. Now we come to the significance of cell division or significance of mitosis cell division. What are the significances of mitosis cell division? Number one, it is responsible for growth. Actually, through mitotic cell division, cell proliferate, cell multiplicates into daughter cells, and through this process, the growth of an organism takes place. So, most important significance is the growth. It helps the growth of an organism. The next one is help in repair and regeneration of cell. The next one is repair and regeneration of cell. This is another type of significance of mitosis, repairing and regenerating of cell. Number 3 is maintaining the constant chromosome number. As we have seen, we have the equal number of chromosome in daughter cells than that of mother cells. So, maintaining the third significance is maintaining the chromosome number of a particular species. Say, for example, in our case, we have 46 chromosomes in our somatic cells. So, 44 cells, chromosomes, we have maintaining the equal number of chromosomes through mitotic cell division. And finally, number 4, in lower organisms, mitosis is the means of reproduction. So, in lower organisms, mitosis is the in lower organism, mitosis is the means of reproduction. So these are the main significance of mitosis. One is growth of the organism, repairing and regenerating of cells. It also helps maintaining the chromosome number constant. And finally, in lower organism, it is the process, it is the means of reproduction for some lower organisms. So today, through this segment, we have discussed the mitotic cell division in animal cell through karyokinesis and cytokinesis, as well as the significance of mitotic cell division. So, see you in our next segment. Till then, goodbye. For testing your understanding of this lesson and more videos, log on to www.tubelessons.net.